going to move on to our next talk here. So our second talk is going to be by Dr. Rovira. Dr. Rovira is a professor in the Veterinary Population Medicine Department, and he's been a large food animal diagnostician with the VDL since 2008. Is that right? Wow.
Right.
So he's been a familiar face probably for many of you, but if you're just new, this is him. He did his DVM and master's training in Spain and completed his PhD at the University of Minnesota. So I'll turn it over to Albert.
Thank you, Matt. Yeah, now that Steph retired, I'm kind of like the old guy. Some of you know that I've been out of the lab for about a year on a sabbatical, and one of the things that I was trying to do was to learn a little bit more about NGS. The more you try to learn about something, you also realize everything you don't know about that. So I'm going to try to share with you some of the things that I learned, some of the things that we already knew before, and some of the things that we don't know yet, but I think could be useful in the future for swine diagnostics. So this is a little bit the organization of the talk. We're going to talk about first about current applications, and some of you have seen this. I'll go fast because we've done this in previous Leman conferences, and we work with some of you on these cases on a routine basis. Then I want to talk a little bit about the state of NGS technologies. I think we're in a critical time right now where there's a lot happening in the different technologies that are out there, and I want to share that with you and see how that could impact in the future what we do for swine diagnostics. And finally, talk a little bit of a few examples about potential NGS applications. These are things that we're not doing yet, but I hope in the future we could do. So if you were in the general session before, Dr. Minion talked about, encouraged us to start conversations with the word imagine. Imagine if we could do this, imagine if we could do that. So we'll do a little bit of that, and like I said, with the situation right now of the different NGS technologies, it's kind of exciting to start thinking about all the things that we could do with NGS. So some of these will be reality, some of these will be potential. Let's start with the current applications. And again, I'm going to go a little fast here because I want to be able to get to the end. What are the current applications that we have right now for swine diagnostics? We can do whole genome sequencing of a known pathogen. That's most of what we do. And once we do that, we can do ID of that pathogen, different types of typing and tracing. And again, that's most of what we do right now. We hope we can predict virulence, predict cross-protection, and predict antimicrobial susceptibility. And we're not quite there. We'll talk a little bit about that, but that would be some of the things that we would like to do. And we also use NGS for discovery, disease investigation and pathogen discovery. Not as much, but things like PCV3, Seneca virus, if you remember, we use NGS to discover those. I won't talk a lot about that, but we do that too. There's other things like microbiome. There's things like cancer research, host susceptibility, a bunch of things. If you think about it, NGS, if you think about the biomedical research in general, NGS is done mostly for other applications, not for infectious disease. Infectious disease is a little part of what NGS can do. But in our world, we use it for infectious disease. So again, this is what we use it most often. I'm going to lower this a little bit, see if it's better. And so, like I said, most of the time we're doing whole genome sequencing of a pathogen that we have already detected. We've already detected this by PCR, right? And we want to know more about this pathogen. What are we doing on a routine basis at the lab? What is it that we do the most? For viruses, we do PRRS, influenza, rotavirus, and then many others in a much lesser degree. For bacteria, we do Streptococcus suis, Glasserella parasuis, E. coli, and many others that maybe are not quite as common. Maybe I should have put staph in here. Is this still too close? How about that? Let's try that. Why do we sequence? What are we trying to do? Well, like I said before, we're trying to trace these cases. We're trying to do comparisons. We're trying to compare this one case with previous cases or other cases that we have, right? what questions are we trying to answer? Is this the same strain that my farm had before? Is this the same strain of the source farm where the pigs are coming from? Is this the strain that the neighbor broke with a week ago or a month ago? Is this a strain that I put into my vaccine or that I use for my inoculation of my pigs? So those types of questions, comparing two or more cases and seeing if it's the same strain or not, however we define a strain. And then we use it as well to inform vaccine production. So very quickly, I'm going to give you an example on PRRS, and then we'll take a look at a couple of examples of very quick examples on bacteria and how we're using this. On PRRS, this is a recent example. April this year, farm was PRRS positive, and we sequenced the R5, and we found out that it was one of these lineage L1C532 that are doing so much damage and that is spreading so much was spreading so much during that spring of 2025. All right, so that system where this is happening, they have seen L1C532s before. In fact, they had seen it 1 year ago. And when we look at this, at the lab level, we have seen several waves, or the last one was a big wave, but several waves of finding this particular strain. In this particular system, they had seen it back in fall of 2024 and spring of 2024. So the first thing that we did was compare it to what they had seen before, the OR5 level. And what happened was this recent one from spring 25, it was acting very aggressive. So is it that it's different from the previous ones? When you look at it, it's around 98%, 98.5%. So this is one of the situations where the OR5 is not really telling us if it's the same or not. It's been half a year or maybe 1 year, and now they are around 2% different. Is this the same virus that just mutated a little bit and then drifted away from the previous one, or is it a completely new one that we introduced laterally and we should be concerned about biosecurity and so on? This is not a good answer from our OR5. We don't want to have a 98. We want to have a 99.9 or 100%, right? Or a 90% to be completely sure of what it is. So because that was not the case, we did whole genome sequencing on these three cases here. And I want to remind you here that when we do OR5 sequencing, if this is a representation of the virus, we're only sequencing this little part of the virus here. So it's very quick, it's very useful, but we have to keep in mind that we're sequencing 4% of the genome. So we decided to go ahead and sequence 100% of the genome to see what the rest of the genome is telling us that OR5 is not telling us. Now, once we did whole genome sequencing, we realized that the new strain, when we compare it to the two older strains, it's around 95% different. So that's a different picture here. So before we had around 98% at the OR5 level. Now we have around 95%. And if you're not used to look at these sequences, that might not seem like a big difference, but it is quite a big difference for strains that, excuse me, are separated in time by half a year or 1 year. And I always like to look at this in a visual way and look at where these differences are located in the genome. So if we consider the original one from May 2024 as the reference strain, here we're representing the differences between that one and the October 24 strains. All this is the genome of PRRS, and then all these lines here, these vertical red lines, represent mutations. So you can see that there are some mutations, but these two are pretty similar. These mutations represent about 1%, less than 1%. So these two, the previous ones, they were within 99% similar. Now here you can see all the difference that you find when you compare initial one to the more recent one. So that's a more visual way to see, okay, what are we looking at here? Are there a lot of changes or not a lot of changes? And also, how are they distributed throughout the genome? If you look at where OR5 is down here, it's no wonder that we were a little bit confused because there aren't that many mutations there. But if you were looking at here or there or there or there, I think you could be concluding that these viruses are pretty different. So after we got this information, there are different ways to interpret this, and there's a lot more information that probably has to come to the table to make a decision. But we could interpret this as the new PRRS strain is different, and perhaps it was recently introduced to this system. Now, this was a good application of NGS for this particular case. Now then we start wondering. We start doing what we said at the beginning, imagine, right? Is a PRRS strain more or less virulent? Imagine if I could sequence the whole genome and just look at certain indicators in the genome that tells me this is going to be, without knowing the clinical picture, this is going to be a virulent one or this is not going to be a virulent one. We're not there yet, but I think it's one of the areas where we want to go to. Is immunity provided by previous exposure or vaccination going to be cross-protected? Imagine if we could provide you, our clients, that type of information with a whole genome. Again, we're not quite there, but this is an area that if we have more research on immunity and we have more whole genomes produced, eventually we should be able to get to those questions. So that was an example of PRRS, an example on a virus. Quick example on bacteria. This is an example of an APP case, and this happened in a system where they're basically negative for APP, except for a few farms where they have it and they had it under control. Well, they started seeing cases of APP in other farms, other part of the system that has nothing to do, different vet, different tracks, different everything. And so they were wondering what's going on. Did we move it or did we get it from something else? Again, we're trying to compare two different cases to see if they are the same, excuse me, or not. So there are, I put this figure here. Usually I don't use this figure, this type of analysis to analyze these cases, but I put it here to remind me to tell you about how bacteria is a lot more complicated. So we were looking at a virus, and the virus we were talking about 7, 10, 15, 20 genes in a virus, and you feel like you can more or less look at the whole virus and have an idea of how it's changing. When you look at bacteria, here we're comparing 5 different APPs, 6 different APPs. All of these combined, they have around 2,300 different genes. And around 1,700 or 1,800, each little line, blue line, vertical blue line is a gene here. So around 1,800 of these are common to all the 6 different isolates that we're looking at. But in addition, bacteria does this thing where they exchange genes. Not all of them have the same genes. Some come and go. So you have some genes here that are common to some of them, but not to the other ones. And you have some genes here that are common to those 2 and not to the rest. So this makes things a lot more complicated. So just so you have an idea of what we're dealing with here when we're talking to bacteria in comparison to viruses. Now, this is what we call the pan-genome analysis. Usually what we do to compare them is we just grab those genes that are common and we just work with those. And that's what I'm going to show you here. What we did here is we saw that when we do the core genome, so that would be the common genes, we saw that they had, these are the previous cases and these are the more recent cases. And we saw that they had a different strain of APP in the newer farms than they had before. And we were able to also predict the serotype of the previous ones, although not the serotype of the newer ones. And then we were able to do other things like looking at potential virulence factors, looking at potential antimicrobial resistance genes based on the whole genomes that we produced. So here we answer the question, is it the same, is it different? It is different. Could we get into more detail on the virulence factors and the antimicrobial resistance? Yes. I don't think we are quite there yet. We'll talk about this in a little bit more. But again, there are things that we could do, and there are things that we wish we could do more, and we're just kind of scratching the surface for those applications. Now, very quickly, just another example just to show you this can get very complicated very quickly. And so this is for Streptococcus suis, and in this case, they had a lot more Streptococcus suis isolates to compare. This was a farm that had, not a farm, this was a system that started having a lot of Streptococcus suis problems. If you remember a few years ago, the serotype 1, sequence type 1 strain that was doing a lot of damage here in the Midwest. Well, what we found is that in most of their farms that they were having problems with CNS disease, this was both pre-weaning and post-weaning, they happened to be all pretty much the same strain. And it was that serotype 1 MLST sequence type 1 strain. What they did in this farm is they started vaccinating with an autogenous vaccine for this particular strain, and things appear to go really well, until they started seeing a little bit more of Streptococcus suis. And what they did is they sent it to us, we sequenced again, and we added it to the previous collection that we had. So this is the result of those extra strains. So the ones in red were additional strains. And just to show you an application of how we use this to make comparisons, and we use this also to monitor the efficacy of control measures, in this case, vaccination. So you can see that there was one of the Strep suis that we found, two of the Strep suis that we found match the previous cases and the vaccine strain. Okay, so those were examples of what we're doing now for viruses, for bacteria. So we're getting the basic question, I think, solved pretty well. Now we have other additional questions that we're not quite getting to those. Now I want to talk a little bit about like I said, the state of NGS technologies, because we're in an interesting moment right now. For that, I'm going to go back a little bit and talk very briefly about sequencing in general and how it started and how it evolved. I like this picture because it puts it in terms of output per run, so how many gigabases that you can get in one run of sequencing, whether it's when we were using Sanger sequencing or when the more common NGS technologies were developed. And what you can see here is that when Ion Torrent, Illumina became more common around 2007, there was a big jump in the efficiency of these technologies to sequence. So we became able to sequence a lot more stuff. And so that's when that was called second generation sequencing or next generation sequencing. And then a little after that, basically these two companies, Nanopore and PacBio, started developing ways to obtain longer reads. I won't get into details, but that was called third generation sequencing. It's a little confusing because that doesn't mean that we stopped doing this. In fact, over the years, you see that we're missing some years here. Over the years, Illumina kept developing and becoming more and more efficient, and other Ion Torrent as well, other brands as well. So short read is still the dominant technology being used. In fact, right now, Illumina, from what I read, has around 80% of the market for next generation sequencing. Remember, this is not necessarily only dedicated to infectious diseases, in general, for all sequencing. Now, what's happening recently? So this kept increasing in the last years because these technologies became more efficient, but they didn't have any big breakthroughs. The machines became a lot more efficient. but the technology didn't change completely. Now, in the last 3 years or so, there's been a lot of new players, input of new companies. There's some patents that expired, and people were able to develop new ideas. And now we have a situation where there's all these new companies trying to compete with Illumina and Ion Torrent and others in that space of short reads. And so that's good for us. For the users, competition is good. They are not just lowering prices, but they are putting out products that are very, very interesting. And I put here 2026 because one of these products is still not on the market and it's coming out next year. But it's very interesting, everything that's coming out. Just to highlight a little bit, a different way to look at the evolution of NGS is tracking the cost of the human genome. And this will remind you that most of the NGS that is done in the world is not for viruses or bacteria like we do in the lab here. It's more for human genomes. It's more for other applications. And that's why they keep track of the cost of the human genome. It's been going down. See, here's 2007, so here it went down dramatically. If you look at the scale, this is a logarithmic scale. So it went down by a lot. And even here, it ends at 2022. In 2024, it was around 600. And in 2025, at least one of the companies claims that was able to lower it under the 100 mark. So a lot of progress going on. And so I wanted to step back here for a minute and explain very quickly to you what the process is, because we're talking NGS, but if I have to summarize the process of NGS, it has 3 parts. One part is sample and library preparation. Another part is the actual sequencing. And what I've been talking about, new technologies, new machines, and so on, is basically on this step where we do the actual sequencing. And we produce a lot of sequences to analyze. And then there's the bioinformatic part. And that's where we get all the data that comes from sequencing and try to make sense of it, try to put together whole genomes, both for viruses, for bacteria. Those are the 3 main parts. Like I said, there's a lot of developments here. These 2 other parts, there's also improvement in these 2 other parts. But what we're seeing now is that as sequencing becomes more and more efficient, it goes down in cost. These used to be more of the bottleneck. Now, bioinformatics and sample library prep are going to probably become more bottlenecks for the whole process. So the fact that this is improving very quickly doesn't necessarily mean that the whole process is going to improve. We have to find improvements in the other parts as well. Now, again, all this improvement that's happening at the sequencing level of these new technologies, they are fighting among them. They are competing, this is a better word, on price, but they are competing on many other things, efficiency, and they are competing on flexibility. Some of these machines are going to be doing multi-omics. Some of these are going to be doing spatial genomics, imaging, things that are hard to believe, but these are going to be really interesting for research. Now, the applications, you have a bunch of applications there. Infectious disease is just one of the applications. And obviously, these companies, when infectious disease is certainly not the main application, they're going to be designing their machines and their products with other things in mind. And it's up to us to try to figure out how to use all the new technology to our advantage for what we need to do. So a lot of improvement, a lot of excitement in this NGS arena, but at the same time, a lot of caveats, and we have to try to figure out how to take advantage of it. All right, so we saw our current applications. We saw the state of NGS technologies and how they are very exciting products coming out. Now I want to show you a few things very quickly about potential NGS applications, how are other people using it, and kind of dream and think about us using it that way as well. So very quickly, for example, this is an example of a group in San Francisco where what they are doing now is instead of doing the traditional diagnostics for meningitis. They are going and doing targeted NGS, which is PCR followed by NGS on their patients to see if they can detect viruses and bacteria straight from the sample. And what they saw, this was an initial study where they didn't have a lot of samples, but what they saw is that with conventional testing, they were able to diagnose 26 patients. If they added metagenomics NGS, or the targeted NGS that I'm talking about, they were able to diagnose 13 more patients. So they are seeing an advantage there to go ahead and do that. Why am I showing you this example? There's a lot of examples of NGS in research where a research group does something, publishes it, looks cool, but how practical that is. Well, these people has been able to make it so that it's commercialized. So they used to do it at the hospital. Now they have a company that does this. And recently they published a study with thousands of cases where they basically found the same results. They are able to basically sequence and find viruses and bacteria directly from the sample by using NGS and cut down on the time that it takes to get the results. So again, pathogen detection. In this case, it would be simply pathogen detection. We're not using it like that. There's a couple of areas where I think something like this could work for us if we were able to put it at a price and a turnaround time that it's practical. There are other companies doing the same thing. So this company also has a lot of documentation about the work that they are doing. It seems to be doing really good. And I put this one here. This one I don't have as much information, but this one is a company that is offering something similar for dogs and cats. So I figured I would put it out here. You might be familiar with them. I don't know very much, but just to highlight that it might be coming into the veterinary world as well. And they seem to have been able to make it work for the companion animal market. Now, one more thought about, or a different example of an application, what's going on with SARS-CoV-2 and whole genome sequencing for COVID-19. I'm sure you're familiar with the variants under monitoring, variants of interest, variants of concern. So there is a global surveillance all over the world for SARS-CoV-2 where people sequence SARS-CoV-2 and put it in a database. And based on the genome, they are able to decide which variants should be under monitoring and which variants should be of interest. If you think about what we're trying to do with PRRS right now, which variants should be under monitoring, we're looking at which variants spread very fast. They don't wait for that. They are just looking at the whole genome, and they are able to predict which ones are more likely to evade the immune response, to transmit very fast, and all of that just looking at the whole genome. Then some of them become variants of concern, and some of them won't. Right now, we have currently 3 variants under monitoring, 1 variant of interest, and no variants of concern that are circulating right now, because some of them, they won't make it. But just looking at the genome, they are predicting what these whole genomes could do. I'm running out of time. This is just an example of how they can map for all of them. They can map all the mutations that they think that are important. And again, imagine if one day we can do this with PRRS, we can do this with flu in real time and give you that information for your particular case. This is what I think your PRRS is like just based on the genome. Matt's standing, so I'm gonna cut it here. I only had 1 left, so not too bad. So again, next generation is evolving very fast. I think this is to our advantage, and I think in 5 to 10 years, we're going to be doing things with next generation sequencing that we are not doing now, maybe even things that we can't imagine right now.
All right, let's thank Albert for his great talk here on NGS.